Good morning, everyone. This is Michele Hughes from Ageless and Timeless. Today, I have Ryan Smith, the founder and now the scientific director. I thought, Ryan, when you were on last time, there was a different title, but we're going with what I see on the internet now as Chief Scientific Director. Is that correct?
Yeah, absolutely. I was working sort of as a CEO prior, but now with all the science and developments going on, I've sort of transitioned full time to research and development. Excited to share that and some of the updates we have today.
Yeah, and you were on, I think, 2 years, 2023, I think is when we first had you on. So there's so much that's happened. I mean, honestly, you've taken warp speed. I've never seen a company do as much in 2 years as TruDiagnostics has done. So we're going to hear all about that today. And the other exciting news is that I did take my second test with Ryan for the TruDiagnostics. And again, I just got the results this morning, and everybody, you'll be so blown away by how comprehensive this test is. And it takes a while to actually to just go through it and to try to digest it. And of course, you need some guidance from someone like Ryan, who or your medical professional who you'd be taking it through to understand all that the implications suggest to you. But there's so much that you can derive from this that it will really reduce the amount of time you have to spend running around to a bunch of specialists if you just take this test. So we're going to go through that today. We're going to make this like a tutorial, very different than my normal podcast. But why don't we start off, Ryan, by just giving us a little background about your journey? Ken Holtorf was on the other day, and he, started chatting about TaylorMade, and TaylorMade was the first company that Ryan actually founded right out of
Yeah.
medical school. So give us a little bit of a history of how this all happened so people know who you are.
Yeah, certainly. It's been a crazy journey for myself, but as you mentioned, shortly out of medical school, I started tailor-made compounding, and my background previously to even medical school had been in peptide and protein synthesis, so sort of making peptides from the ground up. In 2016, which is the first year, we had an idea to start tailor-made compounding to really offer innovative peptides through compounding pharmacy. So we brought a lot of the peptides you might be familiar with to market. Things like the BPC 157, we own the patent for the bremelanotide PT 141. We brought to the market. We brought even thymosin beta, thymosin alpha. The if you name a peptide, we were really one of the first to commercialize it. And as most people know, the peptides are really really popular. But we sort of built that market, but we grew at a really rapid speed. And so we were the fourth fastest growing company in healthcare. We went from sort of me as the first employee to 250 within a little over 2.5 years. So it was sort of incredible growth, mainly because these peptides were so exciting. But we also knew that one of the limitations there was the FDA and some of the lack of data on a lot of these products. And so in the back of my mind, specifically, I was always looking for ways to try and see how these things were helping, but we didn't have time for 20 or 40-year placebo-controlled trials. One of the tools to measure this that really caught my eye during that tailor-made compounding experience was these epigenetic clocks. So these clocks, which are reading not your DNA, but your gene expression patterns, how your genes are turned on and off, to tell you about how you're aging. These clocks have been going on a while, really since 2013 was their first appearance. But really, in 2018, for the first time, they had the first ever interventional data where they were looking at things like metformin, growth hormone, and DHEA to see if they could actually reverse the aging process. And because aging is the biggest risk factor for every chronic disease and death, it became a clock that I was really, really interested in. So unfortunately, finally, the FDA came knocking at our door, and we really had to stop doing a lot of the peptides. But I used that as an opportunity to sort of sell and exit the business to create TruDiagnostic, which really focused on DNA methylation, particularly the DNA methylation age clocks, to sort of quantify aging. And so that started in 2020, and then here we are now, 5 years later, actually to the month actually, and a lot has happened. DNA methylation clocks have come a long way, but the other things that we can do with DNA methylation, and we'll talk about today, I think are equally as exciting.
Well, this is so good. And, I know out of adversity comes opportunity. And in your case, look what happened. Yes, you left medical school because you were disenchanted. That's an adversity at that time. And look what happened with TaylorMade. Then TaylorMade looked like an adversity as the outcome of that company. And now look what happened with TruDiagnostic. So you're like a poster child for creating opportunity out of adversity. And this is certainly something that we all, as entrepreneurs, and people that are looking for answers to health issues, we always look to people like you. And hopefully, others, many of the others that have been on my podcast who have had similar situations, including myself, that,
Yeah.
we have something occurs that looks like it's a terrible event. And
Yeah.
then somehow we look back on it, we say, look what that event did and how it turned my life around. So
Exactly.
kudos to you, Ryan. So let's just do a few definitions for people who are tuning in for the first time, didn't see your last podcast, and many who might not understand these terms. So let's start with peptide. What is a peptide?
Yeah, so a peptide is just a short chain of amino acids. Usually the definition is under 40 in length. So if you're familiar with your amino acids, they make up peptides and proteins of the body, sort of what we call the infrastructure of the body. But peptides in particular can be signaling molecules, acting like hormones or drugs in and of themselves. But they're just short chains of amino acids.
Right. And how do you differentiate between a peptide and a peptide bioregulator? Which, which you and I, how we met actually was through Dr. Bill Lawrence, who was a disciple of Dr. Cavinson from St. Petersburg Gerontology. Unfortunately, he's passed away last year, or 2 years ago, I guess, or last year. And but the work he's done lives on, and so distinguish those two.
Yeah, well, so the bioregulators are still peptides. So, for instance, you think about things like the epitalin or the thymulin; those are still short chain amino acids. Usually the bioregulators are anywhere from 8 amino acids or less, and they're meant to come from natural sources and work on individual products. But peptides generally can be we have a lot of amino acids, and we can have multiple combinations as those peptide chains get larger. And so how those peptides are designed really ultimately influence how they're going to affect the body. But both are the same chain of amino acids, just technically different subdivisions of that category.
And the other distinction is that the peptide bioregulators do attach themselves to specific organs, kind of like the pluripotent stem cells. So, like you can take a peptide bioregulator specifically for your heart or for your lungs or for your bone marrow or your thymus or whatever. So that's a little different, don't you think, than the generic peptides?
Well, one of the things about the bioregulators is that they, outside of a few like thymulin or epitalin, they don't have a lot of published data. And so, those are certainly claims, but I don't know that I've seen anything that would back that up.
Okay.
Generally, the peptides are hitting extracellular receptors, and so generally they might be optimized for a certain organ or another. Like for instance, the epitalin or the pinealon or for the pineal gland, or mimicking products that come from that organ. And so there are certainly, I would say, different categories, but really every peptide is unique and different.
Okay. All right. Well, good. That's good to know. So, okay. Now let's talk about the difference between chronological aging and biological aging. That's some like an ABC of understanding what you're doing at True Diagnostics.
Exactly. It's fundamental. Aging, no matter if we're talking about chronological or biological, it's the biggest risk factor for every disease. But the problem with chronological age is it's not really as informative as we want it to be. We all probably know people in their 70s who look like they're 50 and vice versa. And so it's not really informing us about the biology of how healthy someone actually is. And so biological age is sort of a calculation to tell us more about how someone is aging on a molecular or physical process level. And it's much, much more informative. It's much more predictive of outcomes than just your chronological age.
Today's podcast is brought to you by Epigenetics. From stem cell therapy to lifestyle coaching, Epigenetics is the perfect place to go for all your health and wellness needs. This health clinic is amazing at getting you personalized medicine and wellness that is unique to you. Use the code AGELESS and you'll get 20% off your first service with them. Go to www.Qigenix.com to find out more information about these powerful methods. So the chronological age, just to simplify, is the number of years you've gone around the sun,
Yeah.
and it's just a number. This is why people say aging is just a number, or age is just a number. I mean, not aging. That's not correct. Age is just a number, but biological age is really aging. Is really telling you how old are your organs, or how old are you systemically, metabolically. So that's a huge difference, and I think more and more people are starting to differentiate in their discussions about aging between the biological and the chronological. So I agree with you that chronological age can just be put aside.
Yeah.
When you're looking at, how long are we going to live? It's really your biological age is going to determine your lifespan. So then talk about lifespan and health span.
Yeah, definitely. Obviously, when people think about aging, they think about both, right? How long they will live, but they also think about some of the negative type of events that happen with aging, right? We generally might have our cognitive function decrease, or we might be less mobile and have trouble moving as much. We might have difficulty even sustaining attention sometimes, or we might even have facial aging. All of these things are biological driven processes, so that's really what we're trying to quantify is all of that. But the good news is that health span and lifespan are both pretty linked. Generally, the more that you're improving your health span, the more you're improving your lifespan, and vice versa. There are different. They're giving us different signals, even molecularly when we're trying to predict both. They're slightly different, but ultimately they're very, very correlated. And generally, if you're improving one, you're improving the other.
And so, what about epigenetics? How does so many people are taking DNA tests today, and there's so many companies offering, although my favorite is the DNA company. And as you know, we've talked about Kashif Khan and Tracy Wood, and I've done their tests twice now, just like I've done your test twice. So I have really good feelings about the depth of their testing. But the key thing for people to understand about the DNA company is it's an epigenetic test. And it's not like 23andMe, which is of course now defunct, but where and DNA company never sells your data. So let's talk about the key differences of all these different DNA tests that are out there. But how do you differentiate from your perspective?
Yeah, definitely. So the DNA, right, is a test like you would do for 23andMe. It never changes. You take that test once in a lifetime, right?
Exactly.
But epigenetics is slightly different. What we're basically measuring is how turned on and turned off certain genes are. And unfortunately, these gene sort of activity levels are highly correlated with health and disease. Originally these clocks started by predicting just someone's chronological age. But this data set on epigenetics can now do a lot more than even just your chronological or biological age. We can tell you your risk of certain diseases. We can tell you what you're eating in life. We can tell you what zip code you've lived in based on pollution markers in your DNA. So really, the way that I like to describe epigenetics is sort of a log of everything that's going on in your life. It's sort of all of your behaviors. It's even close psychological relationships. Your gene expression is constantly adapting to try and give you the most optimal health. So that if you encounter some type of illness, you can adapt, or if you have a metabolic dysfunction, you can adapt. And so there are over 29 million epigenetic regions that we would measure in every cell. We call these sort of CPGs. That's where these methylation groups are attached. And so we're sort of measuring the off switch. And so really we measure millions of different locations. And at every single site, we get a number of 0 to 1. The closer it is to 1, the more turned off that gene is. And the closer it is to 0, the more turned on that gene is. And that's not necessarily a good or a bad thing, but we can read those patterns. We sort of always try and joke and say that we're creating the Rosetta Stone of understanding that data set. So what we're trying to do is to use really artificial intelligence with lots of data like that, numbers between 0 and 1, and then also outcome data. So we can try and predict the things that we're trying to predict. So in the case of biological age, we might try and predict that to time until death. So how long do you have to live to create a biological clock? We might though do it to when do people start developing Alzheimer's disease, or do people have Alzheimer's disease, or how much weight might you lose if you take a GLP-1? So this data set is a really, really robust data set that can do a lot of things, but we're really just measuring your gene expression, how turned on or turned off different genes are.
And in those, the term epigenetic really does mean lifestyle. So it means really that you're not given a death sentence by your gene, your what comes out of your DNA test. Because if your genes, like you're just saying, if your genes are towards the turnoff side, but it's a negative gene like the APO4, Alzheimer's gene, but you're not, it's not actively being inspired, let's say, in your body because you're doing a lot of lifestyle things to prevent that. So I think that's a key thing for people not to look at their DNA results as a life and death sentence and then, give up because, oh, I'm going to get Alzheimer's because I have the APO4 allele. That's
Yeah.
not true.
Yeah.
You may never see an activation of that gene, even though your parents gave it to you.
Yeah, exactly. I'm an ApoE3/4, so that's particularly relevant to me. But yeah, no, I think that we've done what we call heritability analysis for things like longevity, and we see that the good majority, probably about 70%, is still based on behaviors versus 30% that might be inherited genetically. And so that's still the large majority, which I hope makes everyone feel a little bit more empowered to take care of their own health. But that is the good point, is that with the DNA test, you generally your DNA won't change across a lifetime. However,
Right.
with epigenetics, it's changeable, and so it sort of allows us to take a baseline, see where we're at, and then ultimately help improve our health by giving interventions and then trying to measure it again to see which direction we're going.
Okay, so let's talk about telomeres. I noticed that the telomeres that you did in the first test with me in 2023, I didn't see in this test. Is that a conscious decision that you made not to include telomere length?
So we still have telomere length, but to be
Oh,
honest with you, we don't love it. And the reason is it's not very predictive of health outcomes. And so a recent study was done by Riccardo Mirioni out of University of Edinburgh, and they really telomere length affects right around 2.8% of the phenotypes of aging. And 2.8% is really, really tiny. Even the earliest epigenetic clocks explain over 28% variance in aging, and the newest epigenetic clocks are more like 60%
oh.
in the epigenetic aging process. And so telomeres are a certain part of aging biology, but they're not always the most predictive of outcomes. And that's really what we're using these tests to do, is to tell us where are we going? What is our trajectory for health? And so even though we do estimate telomere length, we don't prioritize it nearly as much as some of those biological clocks.
Oh, okay. I didn't know that because Dr. Lawrence still requires that as a baseline for the clinical trials for getting into his trials for the peptide bioregulators. That, as you know, he's got about 100 medical professionals and some even consumers in his clinical trials. But the first thing he does is make them take your test.
Yeah.
And I can say this from firsthand experience because I am in his trial, but I think I told you that. But then the second one is the SpectraCell test, which he made. I took twice, but he says he's not so keen on the results of the SpectraCell test. He's not so sure about the efficacy. So he
Yeah.
may be on the same path as you, that maybe that isn't the most important baseline. But what else would he substitute if he didn't do your test, and or does he just need to do your test now because it's so comprehensive?
Yeah, well, at the end of the day, there are a million different measurements for aging, right? And that's because so many different things change as we age. Some people like to just use grip strength, right? Because our grip
Mm-hmm.
will reduce as we age. And so ultimately, whenever we're talking about the testing that we want to use, there's really only two things that are important. And with all the complication that's out there, I always try and just drill down to two important things. The first is what is the most predictive? Because ultimately, again, that's what we're trying to do is we're trying to use the test which gives us the most confidence about our trajectory, right? If we're aging poorly, we want to change that. Um,
Mhm.
so you need a test that's been validated to be predictive of outcomes, right? And so generally what that means is you have to take basically data from biobanks. So biobanks have taken samples for years. So we work a lot with Harvard's biobank, which has taken samples for over 50 years. And so we can actually pull samples from 50 years ago, do their epigenetics, and then see if our algorithms are predictive of outcomes. Our clocks perform very, very well here. We actually can predict death within a 5-year time period with a 92% accuracy. So generally, all of our algorithms are published. They're well validated. Some of our algorithms have been published in over now 250 different studies, showing all of these outcomes with some of the best researchers in the world at your institutions like Harvard, Yale, Duke, Stanford, Columbia. And so there's really, really good data to show that these are the most predictive of outcomes. And that's really ultimately why we're using them. But the second thing that we also want to know is, just getting an idea of where you're at is helpful, but it's not helpful if you don't know how to change it.
Mhm.
The other part is the how. What do we know about changing these markers? And that's been someplace that has been hard to do over the past few years, but now has grown exponentially. We can now offer a lot of different recommendations, and we do that through things like our systems age score, where we don't just now give you an age, we actually give you an age of your different organ systems. So we can tell you
Right,
the age of your liver versus your brain versus your heart.
right.
It gives us the ability to actually take personalized interventions.
So in the tests that you're doing now, do you actually tell people that when they're going to die within 5 years, or is that something too negative to put into a test?
Yeah, we don't put it into the test. The moral implications of that are a little scary.
Exactly.
But we do show that, so the clock that we did to do that is one we created with Harvard. It's called our omic age clock. And it is very, very, very predictive of lifespan. And so instead of reporting on death, we give you just a biological age. So we tell you how it relates to your chronological age. And ideally, your biological age should be lower than your chronological age.
Of
That's
course.
what we want to shoot for.
So what's the average that you see in the tests?
Yeah, so it depends on what group we're looking at, right? In our commercial cohort, the people who take our test commercially, a lot of them do really well on aging, and that's because most of the people who buy our test are health conscious, right?
Yeah.
Already self-selected to be very interested. But if we look at like biobank cohorts, of the general population, generally, chronological age is about average, right? Chronological and biological age. So what we really want though is for everyone to be lower. And so we're trying to get everyone to be as low as possible for as long as possible.
So, okay, so on that first category, what would the average be? How much younger the biological age cohort than the chronological?
Yeah, our commercial cohort, the one that is probably healthier than any other group out there, is on average about 3 years younger biologically than chronologically, which doesn't sound like a lot, but it actually is. It's a big difference. We're talking about reductions in relative risk that are significant in terms of time until death and a lot of others. We even have a website called the Rejuvenation Olympics that we created with Brian Johnson, which is sort of a competitive leaderboard for all of the best agers in the world.
Oh,
So that would give you sort of even the top 10 agers, who's aging the best in the entire world out of anyone who's taken this test.
oh, that's cool. Where, so what would that website be?
So yeah, it's called the Rejuvenation Olympics. I might even just share my screen really quickly
Yeah,
and
sure.
show it to you. But this is the one we created with Brian. Brian started the leaderboard, but has fell off a couple times and now is back on top. So we do this Dunedin pace, which was created with Duke. It is your instantaneous pace of aging. And so really Brian got his as the lowest I've ever seen, to basically zero point 0.5. So for every 1 year, he's only aging half a year in his pace, which is super impressive. We had a couple other celebrities and people on this at one point in time. Another very popular podcaster or YouTuber is Simland as well. But there's a lot of people on here that are aging very, very well. And so this is our leaderboard. Anyone who takes our test is able to add their results here and see if they can make it. We do require 3 tests though to make this leaderboard.
Oh, cool. Can you send me the link to that?
Yeah, certainly. If you
I want to put that in the show notes. So yeah, send me that link. That's really cool for people to see. Well, I feel really good then because I didn't get to do a really in depth of my results, but it did say 10 years
yeah.
difference between biological. So that's a really good result then, huh?
Oh, that's an incredible result. That's something to be proud of.
And I wasn't even that cool about it. I thought, oh no, because at A4M I did a test that told me I was 20 years. So I was a little disappointed, but now after talking to you this morning, I'm feeling much better about that.
Oh,
So,
definitely. Yeah.
well, I promised that we would make this into a tutorial. Oh, one other question before we get into that. DNA methylation. A lot of people don't understand what that is. Can you elaborate?
Yeah, so basically, if people are familiar with DNA, they know we have 4 base pairs of DNA, and one of those base pairs is called the cytosine. And at certain parts in our DNA, those cytosines can become methylated. So we basically just attach a carbon group to them, and that's what DNA methylation is. It's sort of a marker on a cytosine. And again, when that marker is attached, generally that gene is less transcribed into RNA. So it's just less active, it's less expressed. And so that's what we're reading actually, is we're looking at your cytosines in your DNA and seeing how often or not they are actually methylated with that carbon group or not. And the more methylated, again, the less active they are. And so that's really the raw data that we're reading, is we're looking at gene locations and we're saying on a scale of 0 to 1, what percentage of those cytosines are methylated.
And how do you improve that ratio or that score?
Well, then that's the important thing is that sometimes you want that methylation to be very high, sometimes you want it to be low. So
Yeah.
for instance, we're talking about a tumor suppressor gene. We really want that DNA methylation to be really low, right? Because then you're actively suppressing tumors. But if we're talking about an oncogene, a gene that might cause cancer, we might want that to be more methylated. And so it's not necessarily that one is good or bad. It's that we want to basically be able to read the patterns to the optimal health outcome. And that's really what we're doing is by training these algorithms to be predictive of things like disease or death, we know what is bad and we know what is good, and then we can tell you where you're at on that spectrum.
So just personal question to you for a moment. When you did your test, how did you change your results? What were some of the lifestyle things that you did that you can now see have made a difference?
Oh yeah, we've done a little bit of everything actually. So at this point in time, we've done now over 120 interventional trials. So we've done trials in everything from stem cells to exosomes to hyperbaric oxygen to retroviral inhibitors to GLP-1s and weight loss medications. So we've got data on all of these. Personally, I try a lot as well. I'm sort of my own guinea pig, and
Mhm.
I think that we have different things that work for everyone. I tend to be a really big fan of the tried and true methods. And I know that this is not exciting for anyone, but things like exercise, things like stress reduction, things like plant-based diets with, are very, particularly green Mediterranean diets are
Mhm.
excellent. But we've now even published with Yale on over 51 different age interventions and ranked them all together. And so some of the more exotic things that I really like are things like hyperbaric. Hyperbaric is one of the biggest signals that we've seen in healthy populations. I also am a very big fan of things of like the SGLT2 inhibitors, the sort of like your canagliflozin or empagliflozin that make your kidneys permeable to glucose, so you sort of pee out your glucose. I'm a really big fan of those as well. But we've got now a lot of data, and that's really the idea is that we can start to benchmark what are the best interventions for every single person.
Oh, that's so amazing. This is getting more and more finite. And
Yeah, it is.
and
And,
that
and that,
means people can make better choices because it is daunting for a patient to look at these results and say, well, where do I start and how do I,
yeah.
if I'm already living a very healthy lifestyle, how do I get to that more finite level that's going to make the difference in these results? So this is really important, Ryan, what you're doing.
Oh, and that's sort of the vision is that we think that from an aging perspective, we can start to look at all of these aging interventions with the same measuring stick, right? And then be able to say again, the tool that's the most predictive of outcomes is the most reversed by different diet types or different interventions. And so that's really the idea is at the end of the day, that's really what we want to do is give people actionable information to reverse or slow their aging process, which is the biggest risk factor for every disease. And so it's a really, really science-driven endeavor. We're doing a lot of publications, a lot of collaborations, but ultimately we want to take the guesswork out of it.
And are you doing any clinical trials like Harvard did
Oh
and
yeah.
Duke and so forth? I know you're doing joint ventures with them, but are you, as TruDiagnostics, are you taking people in like myself if I wanted to join a trial?
Oh, absolutely. We do this all the time. We've done, studies on a variety of different things. Right now, actually, we just submitted a grant. We were selected as one of the final three applications to get an actual FDA-approved test for aging by the government. And so we're building an interventional trial of over 1,200 people that should go in about a year. And hopefully we'll have the first ever FDA-approved test for aging.
Oh, can you put me on that list?
Yeah, certainly. It's going to be a fun one, and we feel very, very good about it.
I would love to do that. Yeah, and I think being right in the heart of the science when you're looking at all these lifestyle issues. I mean, for example, people like you have on the Brian Johnson website. They're people who are already devoted to improving their health. Maybe not as extreme as Brian Johnson with $2 million budget a year, but and eating his last meal at 11 a.m. I mean, that's for most people that's pretty outrageous and extreme. However, the normal average person that is still showing up on that list is already committed to having a healthy lifestyle. Yet they could still get tests like I did today that
Yeah.
show a lot of areas of improvement. And then you look at the test and you say, but I'm already doing that. I'm already taking D2 and K3. I'm already doing vitamin B complex. And yet the test comes out with some scores that are questionable. So then the question for the consumer or the patient is, what do I do? How do I do the next step to improve what I am already thinking I'm doing that should have produced a better result? So what do you say to those people?
Yeah, I think maybe now's a good time to even go into what you get, maybe
Yeah,
if you actually see some of our results, because
yeah. I
you know,
want that was going to be the next segue, but I just thought it would be good for people to understand that again, like the genetics testing, don't look at this as a death sentence. And just because your result is lower you doesn't mean that you can't. There's still not room for improvement, even though you're already doing. Like I take 1,000 milligrams of D3, and then I take whatever 400, 500 K2, and they're in a combination of I think it's Thorn or Pure encapsulation. So they're good company, and yet still, the test result may have come out a bit lower than what I would have expected, given that I'm already supplementing. So
yeah.
those kinds of things are what I want you to cover when you go over these results. That just keep in mind people don't want to get discouraged.
Yeah, certainly, certainly, and there for our tests, there are really only two tests that we do.
Yeah,
The first test is our biological age test, which we'll be talking a lot about today.
yeah,
And so this is the first one to go over because again, biological age is the biggest risk for every chronic disease, and so we say if there's one thing that you want to prioritize, it's your biggest risk factor, right?
right.
And so ultimately, we have three big aging algorithms that we use here on this one. This is our omic age. Again, we tell you where your chronological age is versus your calendar age. We tell you where you're at within our population, and we can even track those results over time.
Okay,
We can
okay. Well, let you take over the podcast. This is your opportunity now to have people understand exactly what they're going to get in this amazing test, which is not something that most people are used to. They go and do a blood work test maybe once a year, and maybe some specialty test, but never ever is there a test that I know of that is this comprehensive that will tell you so much. So
yeah.
let it be your game right now to tell everybody what this is all about. So go ahead.
Thanks, Michelle. I'll try and make it as quick as possible, just so everyone can get an overview, and then we can talk about how you use it, right? And
Yeah.
the fun part. But yeah, this omic age is one of our algorithms. Again, we can even tell you how your age is increasing your risk of every other disease, because again, we have these outcomes. We've actually measured this in people who have gone on 30 years or 40 years, and we actually know what their risk of developing these diseases were. And then we can actually even tell you what's high or low. So we can actually tell you, what's driving your aging process. So is it glucose, maybe, or insulin resistance, or is it your lipids, or is it inflammation? We can actually start to point out to what's out of range here, and then even make some specific recommendations. So that's what we call our omic age. We develop with Harvard. We also have our symphony age. This is one we developed with the researchers at Yale that actually gives you the age of each organ system. So your lung versus your metabolic versus musculoskeletal. And again, this helps us give more precise recommendations. Someone who's a smoker, for instance, might have really high inflammation, or they might have really high lung age, but someone who's maybe overweight might have metabolic aging. And so we can actually now start to differentiate for individual system recommendations. And so this was a big step forward, and we just had this now. We've only had it for about a year on the platform. And then, we also have probably everyone's favorite, which is the Dunedin pace. And so that's that rate of aging we had on the Rejuvenation Olympics website that tells you how fast you're aging right at this moment. The reason that people like this one the most is because it's one of the most well validated, but it's also the most correlated to health span related metrics. So a lot of people think, I don't want to live, till I'm 120 and just have a really poor quality of life. And so if you really want to maximize things like your IQ and mental processing speeds, even your facial aging is really tied to this metric. And so for people, they really like this. It's also more responsive to change. And so again, we want this to be as low as possible. Well, again, Brian's is the lowest we've ever seen at 0.5, but as long as you're below 1, that's really what we want to see there. And so those are some of the biggest aging metrics, but we do a lot of other things on this report too. We can tell you all about your immune subsets, to see if you have any immune deficiencies. We do tell you your telomere length here as well. And we can even do things like telling you how much you smoke or drank across a lifetime based on these markers. And so that's really what our true age report looks like. And it's really meant to quantify that aging process. And again, there are a lot of different age tests out there, but these biological clocks are all validated. They're all published. They've been compared in interventional trials, so you know what changes them. And they provide a lot of really science-based feedback for you on the aging process.
I apologize. I didn't see the telomere length in my test, so I did say that I didn't see it this time, but I guess it is there, so I'll go back and
Uh,
look at that for,
Definitely.
for the one you
Yeah.
sent me. Okay.
And yeah, but the next thing I want to introduce is actually something a little bit different. It's our other report we actually call our True Health Report.
Mhm.
If you don't mind, I'd love to explain this a little bit more because this is one of the biggest surprises that we had at our company. Whenever we wanted to create the omic age, we noticed one big thing, which is if we wanted to create the best aging clock available, we really needed the most data available. And so we really wanted to measure everything we could possibly measure in this cohort. And so in order to do that, we have this classification system we call the multiome, and it really starts with genomics, right? What is that DNA sequence? Then it goes to epigenomics, and that is again what DNA is expressed, what's turned on and turned off. That then creates RNA, which is transcriptomics. That RNA then goes to the ribosomes to create peptides and create proteins. That's what we call proteomics. That really creates the infrastructure of the body and then creates metabolites. And so there we're talking about hormones, we're talking about neurotransmitters, we're talking about all of the different chemical exposures you might have in your environment, like PFAS chemicals or even microplastics. And then ultimately you have the outcome, what ended up happening to the person. And so we took all of this data that we possibly could and measure this in our Harvard cohort. The problem is it's really expensive to measure all of these different things. And you also need plasma, you need phlebotomy. And so we basically asked the question, can we just with methylation predict all of these other values? So can we predict your hormone levels? Can we predict your neurotransmitters and cortisol? Can we predict your clinical function measurements like your VO2 max or your HbA1c? And the answer was yes, we actually could. We can actually do this with 1,700 different biomarkers just from DNA methylation. And one of the coolest things about this is that they actually tend to be more predictive of outcomes than even your classical biomarkers. And so this is when we created this report we're calling True Health. And we actually think that in the next few years, this will replace all of your clinical blood testing. There's
Mhm.
no reason you'll need to go to a LabCorp or Quest to get your bloods drawn because we can actually report on all of these different things that you would see here. In terms of categories, we do a lot. We do all of your vitamins, so all of your B vitamins, your A, D, E, K vitamins. We would do all of your amino acids, like your, taurine or glutathione, which we know is a great longevity-specific amino acid. In addition to that, we can do all of your antioxidants, like your carotenoids. We can do your fats and cellular membranes, like your omega-3 ratios, your omega-6 ratios. We can even tell you about, your NAD biomarkers, your lipid particle sizes. We can get so much information now from just a single drop of blood, and running all of these different algorithms on it. And again, they are more predictive. 62% of the time, they're more predictive of the classical biomarker. So, for instance, if we're trying to predict cardiovascular disease, a lot of people look at blood pressure. But our prediction of blood pressure through epigenetics is actually better than even the measured blood pressure that you get in clinic in terms of predicting cardiovascular disease.
And why is that, Ryan?
Yeah, so one of the reasons we think that this is the case is I always like to use a Steve Horvath quote, which is the another potential benefit of using DNA methylation-based biomarkers is they represent a longer average concentration. So the best similar example is HbA1c and fasting glucose, right? Our fasting glucose can vary all throughout the day, and if we get it at a single point in time, it might not be indicative of our overall health, right? We might have just had a big glucose meal, right? And our
Mhm.
glucose is super high. It doesn't mean we have diabetes. It just means that our glucose is high at that moment.
Right.
HbA1c is a 3-month running average of our glucose levels. And so it gives us a much better idea of what our overall glucose is. And so the same is true with these DNA methylation biomarkers, is that we're getting a longer average and therefore it's more predictive of outcomes than even just a single point in time measure. Yeah, again, we published with, a lot of really good data here I can show, but we published with Harvard that, again, 62% of the time, our biomarkers are better than the classical measures. But again, we don't have to get whole blood. We don't have to, so we don't have to go to a phlebotomist. We get over 1,700 of these biomarkers from a single drop of blood. And so the scale and accuracy and predictability of these things is sort of incredible. And that goes into this report where we actually tell you, again, where you're at, if your HDL particle size is high, if it's low, and then we try and give you recommendations on how to improve that.
Mhm.
And some of the things we can do here are pretty incredible. We can tell you even what you're eating based on these different metabolites, and then even suggest ways to improve that. I'll show you maybe one more example if you don't mind. One of my favorites, which is that we can actually, again, look at what you're eating based on the metabolites in your blood. So, we did this by taking self-reported food behaviors and linking them to metabolites. And then we can do a network analysis where we can actually tell you what metabolites are linked to what foods. And so we can actually tell you what you're eating now based on this testing. And then we can actually apply it. So this is our omic age algorithm. The things here that you see on the left are the foods which are best for this aging versus the
Mhm.
foods on the right, which are worse. And so we see things like fish, eggs and mushrooms being really good for the aging process. And on the other
Mhm.
side, we see things like sweetened foods and beverages, cereals, and processed foods being really bad, which again is pretty intuitive, but we can actually read this all through your DNA methylation profiles. And so it's really incredible the amount of information we can get from this. There are multiple case use examples, but again, just from a single drop of blood, we can tell you all of this information.
Oh, so Ryan, was all that in the test that you sent me today?
Not yet. So the food and diet things will be on our future tests.
Okay.
And again, we're even adding the ability to predict disease. So we'll be able to tell you your 5 and 10 year risk for things like Alzheimer's or cardiovascular disease or diabetes. But right now, it's not commercialized. It needs a little bit more research and development before we roll it out.
And what about hormones? I don't see I know that you're going to be focusing on hormones at some point.
Yeah, hormones are obviously super important as we talk about aging. Unfortunately, hormones tend to be one of our big weaknesses at the moment. People who take exogenous hormones sometimes mess up our readings. And so right now, hormones are not on our list of things. We can predict some hormones like thyroid, some like cortisol and cortisone, but we can't do sex hormones right now,
Yeah,
like testosterone or estradiol.
I saw that.
Mhm.
I was disappointed, but I say, hey, they've done so much else. Uh, but I did, it was a question. It is a question because the hormones are the superhighway of your
Yeah.
body, of your health. So, but you do focus on mitochondrial function, and I noticed though that you kind of dodged the issue of metformin and some of these that you talked about earlier that you did at TaylorMade. Is there a reason for that you're not giving recommendations for the pharmaceutical side for aging, for anti-aging?
Yeah, so we're building those data sets right now. A lot of people like to recommend metformin for aging. We're not as certain. The data that we've seen doesn't look as overall compelling, I think.
Mhm.
We've done actually metformin in an HIV population, and it was phenomenal. We had anti-aging reductions across the board, but in normal individuals who don't have any blood sugar related issues, we don't see the biggest change with metformin.
Mhm.
Even with the GLP-1s. If you're already pretty healthy and you don't have metabolic issues, we don't see a big change with the GLP-1s. But if you are metabolically unhealthy, we see massive aging reductions in the GLP-1s.
Mhm.
And so, we've looked at everything from rapamycin to, metformin we just published a study on statins even.
Mhm.
And so we're getting all of this data, but right now we're hesitant to recommend, drugs for aging that have been approved in other areas.
And what about the peptides? So, I noticed in the test that this morning, just reviewing it, and you made some recommendations of supplements to take, but I didn't see anything with the area that you're the most familiar with, which is peptides. So, is there a deliberate reason for that?
Yeah, it's all based on data. So we will not recommend anything that doesn't have a clear, I would say, data publication relationship. And unfortunately, with the peptides, I'm a huge fan of the peptides. I couldn't say enough positive things about them. But unfortunately, they don't have very much studies or data. And a lot of that is because these things are oftentimes their patents have expired, so there's no reason for people to invest in this research. And that's really sad. I've seen, most people know even products like, CJC or ipamorelin is these growth hormone products. But ipamorelin has been bought by GlaxoSmithKline and really left and abandoned right on a development pipeline. And so unfortunately, there's not a lot of economic incentive to have these things studied. And so unfortunately, at the moment, there's not a lot of data, and therefore we don't make those recommendations without very clear data.
Yeah, so this is what's happening then, just similar to what happened with Apple and Google and the others, where they would acquire their competitors and then just put them out of business, basically. Is that really what's happening now with Ipamorelin and so Smith Kline owns them now?
Yeah, so they're all owned by a variety of pharmaceuticals. SS31 is owned by Stealth Biopharma. You've got the thymosin alpha or Zadaxin that's owned by Cyclone Therapeutics again, and they've really all stopped their development and spending. And so even though they're great products, it takes $2.3 billion to go to the FDA and get an approval of a drug. That's a lot of money, and very, very, very few companies throughout the world would have that amount of money.
Right.
And so just because I think there's a different threshold, but unfortunately, we don't have data for those things. We're always trying to get some of that data from our commercial cohort, people who are already using them.
Hmm.
And we can do cross-sectional analysis, but we can't do a placebo-controlled trial because it's really not sponsored or approved in the typical formats.
Is this the same for the semiglutides?
Semaglutide's different. That is owned by Novo Nordisk. It goes off patent in 2027, and we've actually published two studies on the semaglutide, one in a healthy population and then one in an HIV population. But we have even created an epigenetic predictor of weight loss response, so we can actually tell you if you would be a good candidate to lose subcutaneous tissue with that different intervention.
Is that a test that you have to take separately to get that info, or is that not? It's not
Yeah,
available yet.
it's not available yet. We haven't commercialized it, but we have published on it.
Okay. And you saying that there is going to be a test that you can take to tell you whether a semiglutide is going to be beneficial for you or not.
Yeah, and even beyond that, we'll be able to tell you if you're going to lose muscle or visceral adipose tissue or subcutaneous, and we'll tell you if you have side effects like GI side effects or nausea. So it'll be almost like a companion diagnostic to be able to use to recommend which GLP-1 you might want to be on.
Wow. That's just absolutely amazing, given how widespread and how abused
Mhm.
this semaglutide, the GLP-1s have become, because people just, one, there's so much obesity in this country that and diabetes, that people just want to find anything that will make a change, and sometimes they don't do the research.
Mhm.
The doctor isn't as thorough. This just gives them the new prescription and say, come back when you've lost your first 10 pounds, and
Yeah.
forgetting about the fact that sarcopenia is one of the biggest side effects. Not only that, but there are many people reporting other serious side effects like, gastrointestinal and nausea and all that. So this is going to be amazing. I mean, so what's the timeline?
Yeah, I think probably just in 6 months or so we'll be able to have it, and to be honest with you, we might even be able to add it for free on all of our reports.
Oh my goodness. Oh Lord.
Yeah,
What is
and
the cost? What is the cost of the test as it is today?
yeah, so generally, I think we do each of our reports for around 4.99 direct to the consumer. However, I think we always try and give some discounts on that, and I think that if anyone's watching this, they can use the code ageless20 for 20% off that.
So it would be 500, let's say, for each test, so $1,000, so they could get it for 800.
Yeah, well, and actually the combo is we do offer for 8.99. Again, if you're getting
Oh,
20% off that,
oh,
it'll be even cheaper. Mhm.
so it'd be about 700. Yeah, wow.
Yeah.
Everybody listening, I hope, and watching today, this is like amazing. It'll be one of the best $700 you've ever spent for managing your health. I can say that with all, and I'm not even an affiliate with you, or there's no financial considerations here. I say this only because I'm a believer and because I want to help people. And really, I'm going to send this test to my doctor. You know who my doctor is, and say, why am I taking my LabCorp test in August, my annual blood work, when you've got this test? Do I really even need to go
Yeah.
in? Is there anything missing on this test that isn't on the typical blood
Yeah.
work?
Outside of hormones, not really. That is at the end of the day, the one thing we're trying to add to make it a comprehensive test. And again, even when we compare them head to head, we know that these are actually better than even your classical blood work.
Have you had any repercussions from LabCorp or Quest about the fact that
Not yet. We just published the preprint in December, and so this is still very, very new. But we're really excited to get this more widely used and validated. I think that in the next 3 months, we should have 6 or 7 validation studies coming out showing just how good and predictive these are.
so if people just go and do their hormone tests, that would be all they would need? It just because I last time I did my blood work, they took 20 vials of blood, and she does really comprehensive lab work. So, well, I guess they'll do it this year just to compare the
Yeah.
lab results with your results to see where there's a discrepancy. That might be an interesting.
Yeah.
Do you think there will be discrepancies?
Well, probably, but again, the only thing that really matters again is how predictive it is of outcomes, right? And
Yeah,
so again, if you were going to compare your glucose and your HbA1c, they wouldn't be highly correlated because one's a point in time, the other one's an average. Ours is that average, right?
right.
And so there might be some discrepancies, but again, that's why we publish, that's why we have this data to show that our markers are effective and validated.
And just for everybody to know, all you have to do is get a prick of your blood on your finger, and you put it on a card, and you fill in the little circle of the card. Do you have a picture of that
Oh,
anywhere?
unfortunately, I don't have one readily available, but
Okay,
oh, actually,
I have it in my photo gallery because I
yeah,
took a picture when I was at the lab, and yeah, there it is. You have it, right?
yeah. It's just a small finger stick circle right there. We just
It's unbelievable. I mean, compare that to 20 vials of blood and
yeah,
how you
exactly.
feel afterwards.
Yeah.
Well, Ryan, how do people reach you to get the test? That's really important.
Yeah, so TruDiagnostic.com is the best place. You can order the test directly. We do oftentimes recommend taking it to your provider to help analyze and interpret.
Yeah.
And if you need help with the recommendation of provider, feel free to reach out to us, and we're happy to do it. You can also find me on LinkedIn. Really the only place that I'm active. But feel free to reach out if anyone has any questions or needs to be connected to a provider.
And not only that, but Ryan has very graciously done a number of podcasts. I was watching you this morning on Sam Tejada and Joy, Dr. Joy Kong. I think Kong is her name. Anyway, those were the two that I just came up. But if you Googled your name under or YouTube, you're very generous with your time and really helping people to understand more about the not only the science but the just the efficacy of these results that can save people so much time.
Yeah, I'm
So
a huge believer in preventative health, and I think this is really true preventative health.
yeah. So well, before we close, Ryan, what's next?
So, I
Other than what you've already talked about, I know all these incredible advances that you're making, but what else do you have in mind for True
yeah.
Diagnostics?
Well, I'll tell you at the end of the day, my vision is to have one diagnostic you do twice per year that can report on your risk of every disease, report on your risk of lifestyle things that might be a problem. And I think ultimately we're going to get there. One of the ways we're getting there is creating the biggest combination of multi-omic biological data. And ultimately we're going to start to get a lot of FDA approved tests across a lot of indications. So I really do think that with one drop of blood, we can start to do some really great, low cost, highly accessible healthcare. That's the envision. How we go there, we'll start with aging and then expand to other disease.
And then you were saying in the letter that I got this morning that to do it every 3 months. So you're saying now twice a year, but that's a goal. But you're for now, is it still 4 times a year?
Yeah, you can take it at really whatever interval you'd like. We've even seen changes in as little as 3 weeks with things like our hyperbaric studies.
Right.
But I think that my goal is to get insurance reimbursed twice a year by Medicaid, Medicare, as well as any insurance.
And is it now available for insurance?
Not,
Not.
and we're working on that right now. Unfortunately, the FDA doesn't have a recognized aging as a disease, but that's really what we're hoping to change.
Yeah, no, I had Eric Verdon from the Buck Institute, the CEO, and we got into that discussion about is aging a disease, and or how do you define aging? And so he said no, his contemporaries say it's not a disease. I think he's coming more from that allopathic
Yes.
model for that, even though the Buck Institute is becoming much more of a functional medicine. But anyway, he said no, it is not a disease. So what is it then if it isn't a disease?
Yeah, this progressive deterioration, it goes back to the definition of a disease, but I think ultimately it doesn't matter how we call it a disease or not. If the FDA can prioritize the usefulness of a diagnostic, that's
Hmm.
what we care about. And they are. In that grant I mentioned they're applying for, it's called something a little bit different. It's not called aging, it's called intrinsic capacity, but
Yeah.
ultimately it's the same principle.
Well, hopefully, with Robert F. Kennedy and the change of the direction, that you're going to have a lot more support for what you're doing, don't you think?
I hope so. Fingers crossed. Unfortunately, a lot of our research collaborators are starting to lose funding in this environment, which is
Yeah.
not something I love to see. But I'm hoping the NIH can still keep their funding, and it's so important for everything that we do.
No, I know Eric was saying the exact same thing that about 50% of their funding is NIH, and that's being curtailed. So yeah, we've got to find a balance because I mean, obviously, RFK recognizes that being in the 80th percentile of the nations in the world, and particularly the progressive nations like the United States, we should be number one for the amount that we spend in healthcare. And look at our track record; it's really quite deplorable.
It is. We need improvements, and again, but this systematic framework is the way to do it. So I'm hoping that we'll see some change. But ultimately, if there's one takeaway for anyone listening, it's treat aging as something you can change and modify.
And
It's
the epigenetic side.
yeah.
Ryan, I have to tell you before we close, you are one of my big heroes.
Thank
And
you.
as I got into this world, I mean, I've been passionate about functional medicine for many, many years. But when I really put my 2 feet in with six years ago with the podcast, and now I write an article for Top Doctor magazine. monthly. And so this is, why are you my hero? Well, because I see someone really young who just took a risk, and truly, I mean, this was a risk. You left medical school, you gave up the traditional financial model, and you took this risk, and look where we are today for all of your work. And yet you're so young, so we have so much more to anticipate with all your incredible energy and your wonderful spirit and positive attitude. So thank you so much for doing all you can for all of us.
Likewise, Michelle, I get my head buried in these papers and books, and sometimes it's hard to explain all the work we're doing and the vision that we have. And so I thank you for having me on and letting me tell the story.
Well, you've done this for many other podcasters, but I take great honor that you have been on ours twice. So, all right, Ryan. Well, you have a wonderful 4th of July, and thank you again. And I'll see you probably in December at A4M.
All right, let's do it. Thank you so much, Michelle. I appreciate
Okay.
it.